Hello, everybody. Good evening. With a show of reaction, kindly let me know if you can get my audio. Thank you. Thank you so much. Karibuni sana for this session. Let's remind our colleagues to come in so that we begin together. In the meantime, kindly just introduce yourself in the chat box, name and your area of practice so that we see who is in the room with us. Yes, thank you. Thank you so much. I can see some good reaction and some good representation. Dr. Caleb Ochieng from Turkana, karibu sana to the call. There's Dr. Michael Tushati Pirdai, Kajiado County, karibu sana. Nadab Omondi, Amolo, karibu. Good to see you in the room. I see there's a representation from Lamu County, Elgeyo Marakwet. We have good numbers from all over the country. Feel welcome. We would be beginning in a few. So just to, again, invite you to share with your colleagues, remind them to join us so that they don't miss the discussion. We'd like to do this together. My name is Dr. Silvanus Manyala, and I will be introducing a few ground rules to you, and then, of course, hand it over to the moderator of the night, who is going to give us some context about the session. And of course, it's a series, so more about what is in store for us as well. And he will also be having the opportunity to introduce the speakers to you so that you get to know who you will be hearing from. But just as a reminder, as you can see on the screen, this is a webinar that is being delivered to you by the Innovation, Research and Digital Health Sectoral Committee, and it is titled Demystifying Research, Understanding Clinical Research. We have Dr. Mangeni Koba and Dr. Kelvin Jerry who will be speaking, and of course, moderation by Dr. Victor Mulelo. Next slide. Next. Yes, thank you so much. So again, this is how it would look like. We have opening remarks after my part in the next 3 minutes, and then speaker presentation. Thereafter, we'll have a session for discussion and engagement in a Q&A segment, and then we will close afterwards. Next. So just as a reminder, this is a standard practice. We would like to let you know that you are muted for this session, and you will remain in that state until the Q&A, maybe when we will have the opportunity to give you the floor to speak. And we encourage you during this period of the delivery to engage in the chat section, just as I had instructed earlier. If you have questions, kindly raise them in the Q&A pod at the bottom of your window, you should be able to see some icons. If you do not see them comprehensively, there's a more icon with 3 dots. You can click on it and you'll access the Q&A, where you raise questions and then they will be responded to during the Q&A segment. And of course, we want to, of course, inform you that we'll have the recording of this session available on our YouTube channel. So just remember that you can always look back and listen, and of course, just see what was discussed. This session is CPD accredited. At the moment, we are still following up with the Pharmacy and Poisons Board, as the event is not listed, but it should be in the next few hours or maybe next few days. And we will communicate the same to you via email, of course, with instructions on subscription, and of course, the deadline for claiming. Remember, attendance is important. We will only award the tokens to about 75% of attendance. Maybe at this juncture, I'd like to hand it over to the moderator of the session to take it up. Karibu sana, Dr. Mulelo, over to you.
Asante sana, Dr. Manyala. As you've been told, I'm Dr. Victor Mulelo, a health policy and systems researcher at the Strathmore University PRISM Center, and I'm committee member of the PSK Innovation Research and Digital Health Sectoral Committee. So where did this all begin? The Innovation Research and Digital Health Committee started by asking the question, where precisely are the gaps in how the pharmacy profession engages with research? And after conducting a desk review and rolling out a questionnaire, what came back was a mapping of the priority knowledge gaps that exist in our profession. Our response is a two-pronged approach. The first is a series of webinars that works through those gaps one at a time. And the second is thought leadership through writing of commentaries to be published. The first webinar series was delivered on the 30th of July, which covered understanding health policy and systems research. And tonight we have series 2, which is a deliberate progression to help you guys understand clinical research and how it differentiates from health policy and systems research. Our first speaker of the day will be Dr. Kelvin Jerry Wandera, who is a principal investigator and clinical trial pharmacist of record with over 15 years of experience in clinical trial research. So you can see you guys are in safe hands when it comes to clinical research. He has extensive expertise, studied leadership and research operations. He has implemented industry-sponsored, investigator-initiated, and global clinical trials across HIV, tuberculosis, malaria, vaccines, medical devices, among other therapeutic areas, working collaboratively with different sponsors, clinical research organizations, regulatory authorities, and site research teams to deliver high quality, ethical, and scientifically rigorous clinical research. I hope in the course of this presentation, Dr. will also share with our audience the amazing work that you are currently doing, because I know for a fact it's very impactful. So, karibu Daktari.
Thank you very much, Dr. Mulelo. And I want to also say thank you to the Innovation Research Committee for this chance so that some of us can say what we normally do under the silos. Without wasting much time, we would like to check on the session outline, whereby we are going to check on what clinical research is, but basically we'll start on the foundations, then the clinical research designs, then ethical and regulatory pathway. Basically, this is to ensure that you as a pharmacist are able to tell, like if you are to be given a protocol today and you need some approval, where do you start? And if you are given a protocol today, what clinical research design is in that protocol? And basically the clinical research, what it is and what it entails. I hope I'm very audible, and let's start on the next slide. So when we start on what is clinical research, this is a basic thing, and it's just the systematic investigation of medicines, devices, or intervention involving human participants. So the basic thing we are looking here is for safety, efficacy, and effectiveness of what we are going to give you as an intervention. Does it work? Is it safe? And basically, we would like to say that the clinical research is evidence engine behind every medicine you dispense as a pharmacist. So that paracetamol, that diclofenac, that amoxicillin you do dispense on a daily basis is anchored through this clinical research. And according to the National Institute of Health, it should be able to answer the question that it's a trial, it has human participants, it is a prospective assignment to an intervention, and it has a health-related outcome whereby it helps you to cure, prevent, or manage a given disease condition. We can move to the next slide.
Doc, could you kindly increase your audibility?
Is that okay now?
Yes, that's perfect.
That's perfect. So I can repeat. For clinical research, the basic thing we want to know is a systematic investigation for a drug, a device, or an intervention, and it should involve human participants and also designed to generate something on safety, efficacy, and effectiveness. And as I had said before, the NIH, meaning the National Institute of Health, this comes from the American. It gives us some definition whereby it should involve human participants, prospective assignment to that intervention that you are giving, and it has an evaluation of an effect and a health-related outcome. And basically, we say for every drug you're dispensing, wherever you are as a pharmacist, it comes out from a clinical research setting, be it the paracetamol, the amoxicillin, or any other drug. Next. So phases of clinical research, these phases are known like by everyone. And the first thing you should know, there are 4 phases, and each phase is unique. So for a pharmacist engaging in research, you should know all these 4 phases. And phase 1 purpose is to evaluate safety, tolerability of the agent or the intervention. It is worth anchoring that drugs' life in each design tend to follow up. Phase 1 is that safety, tolerability, and it's often called also first in human. And in this, I've given an example of an investigator-initiated study that is phase 1, which evaluated safety of adjuvant self-administered vaginal 5-fluorouracil cream following cervical intraepithelial neoplasia grade 2 and 3 treatment among HIV-positive women. So in this trial, we were using this drug to manage this cervical intraepithelial neoplasia, which was at grade 2 and 3. And then in phase 2, the most important thing you have to check is evaluate effectiveness, and safety assessment continues. And an example of this study that is in the pipeline is a phase 2 study of adjuvant 5-fluorouracil following thermal ablation to improve HPV treatment outcomes of women in Kenya. So this means phase 1 was done, now we are on phase 2. And still we are looking for effectiveness. So phase 3, it works better than does it work better than the current standard? And in this phase, you confirm effectiveness, you monitor side effects, and then there are those studies that will tell you we compare it to the placebo or we compare it to the standard treatment. And the example is that we have a phase 3 double-blind study for RTSS vaccine for infants who were having malaria. So I would also add another one here, whereby you have read protocols and seen vaccine trials whereby you use the vaccine itself, which is the active ingredient, and a placebo, which is normally used in most cases is normal saline. So that is where the catch is. And we will dive in further and know when you use a placebo, when you use a standard treatment. Also in phase 3, you can come up with a study whereby you need to evaluate, for example, a TB drug that is new in the market. You will compare it to the standard treatment. And you'll find out that for TB patients, you cannot give them placebo. So you need to do one arm to have the active participants who are receiving the new intervention versus the other arm that will receive the current standard regimen. And then in phase 4, we are dealing with long-term impact. And in this can give you the optimizing RSV pragmatic trial, whereby this RSV's vaccine is readily available in European countries. So we give it to pregnant mothers when they are at 32 weeks. And then we follow those mothers until they give birth. And after that, we follow them until those children grow up to around 4 years. So in this case, we are able to tell, do they get respiratory infections despite them getting the RSV vaccine? Next. So we also have to know who is a clinical trial sponsor. Clinical trial sponsor is always an individual or a company or an institution that will take responsibility of study, initiation, management, and financing. Basically, these guys provide the finance you need to run the trial, and also they do close to close GCP compliance. They also ensure quality and they protect participants. These clinical trial sponsor are basically the big pharma companies. And in this, we mean companies like AstraZeneca, GSK, the Janssen, the Pfizer, and the Sanofi. These are your big pharma companies. They provide oversight to the trial. They are the ones who are held accountable if something goes wrong on a trial. And that is the first place where accountability lands when you are in a trial. So an example of them are listed down here. There was a link here. Maybe I will share the link in the inbox on Transrelate, whereby it will give you a lot of these trial sponsors and you will be able to know them. And basically, when it comes to pharmaceutical jobs, they are also at the forefront giving us jobs, whereby I think my next counterpart will also have to explain that. Next. So another thing that you also need to know is the contact research organization. And you should know that sponsors do not run clinical trials by themselves. And a lot of work is contracted out to contract research organizations, abbreviated as CROs. These are independent organizations that the sponsor will contract to do specialized research services. And some of these services are like clinical trial management. They'll come on site also to do monitoring. They'll do what we call site selection. Site selection involves checking if your site has got adequate staffing, adequate equipment, adequate regulatory compliance, which is local for you to hold or conduct a clinical trial. So all these are done by contact research organizations. And then this contact research organization also will help you in regulatory affairs trial submission. While you are making a submission of a clinical trial to an ethical body or to the regulator, in this case, the Pharmacy and Poisons Board, these contact research organizations will help you prepare those dossiers that are required for submission, and then they get to be reviewed, and after reviewed, your study is also approved. So some of them I know you have come across, IQVIA, Thermo Fisher PPD, the biotech, and in Kenya we have this CleanWin that also provides a course for you to learn about how to become a monitor. And another one also found in Kenya is the Croissants Clinical Research, and another one also found in Kenya is the ACE Research. So these are locally based here in Kenya, and they'll be able to assist you know how to navigate your way as a pharmacist in one of the career pathways. Next. So for clinical trial sites in Kenya, here, what we basically see are sites that are able to conduct clinical trials. And as I've previously mentioned to you, when a CRO comes to a clinical trial site, they'll have a list of what to gauge you as a site so that you're able to do a clinical trial. And in this case, these are what we call feasibilities. These are forms or questionnaires that a site is given so that they can fill in their data. And this data is all about how you'll get participants, how the site is interested in doing such clinical trial X or Y. You also fill your site capabilities, staff with expertise. If need be, how can you fast scale the number of staff to like do a clinical trial that requires a lot of numbers to be enrolled? And some of these sites in Kenya, I can say Kemri is the first one, and it has substations everywhere. There's a station in Nairobi, Kisumu sides, Mombasa sides. We have a lot of Kemri stations. Then you have Kavi, is situated in KNH Nairobi. We also have Ampath. We have Pwani University. We have our own impact research here in Kisumu. Also a sister site in Kisumu, the Nyanza Reproductive, and also another sister site in Kisumu called Vibri. Also, the Aga Khan does research. So if you are to look for jobs on research or mentorship or apprenticeship, you should approach such institutions. And this will be explained further also by Dr. Kuba. So basically, these are the trial sites in Kenya, but not also to forget, this is just a small list. We also have these referral hospitals within our counties that also conduct clinical research. So you should also take note of that. Even the National Teaching and Referral Hospital do conduct research. Next. So when we go to clinical research designs, we are talking about randomized controlled trials. In these randomized controlled trials, participants are randomly assigned to an intervention or a control arm. And this is producing an biased estimate of the treatment effect. So to me, the best definition I can give you of randomization is like flipping a coin where you get a head or a tail. And I also think you also know randomization also takes place when Arsenal is about to play Manchester United, whereby they come at the center of the pitch, they toss a coin, they say this one to the left, this one to the right. So in such a similar manner, you are able to tell this one has gone this side and this one has gone this side. So an example of a randomized controlled trial you can see is a phase 3 randomized active control double-blind study to evaluate efficacy and safety of oral MK-8527 once a monthly pill as HIV-1 pre-exposure prophylaxis in cisgender men, transgender women, transgender men, and gender non-binary people who have sex with partners assigned male as sex at birth. So in this trial, if I can dig deeper, it is a HIV trial, and you can ask me, why are people getting randomized? And you can ask me, what is the intervention? So on one side of the arm, the intervention is the MK-8527 oral pill and the standard of care, which is the FTC TDF. So in this trial, we have two groups. Group 1 will get an active tablet of MK-8527 to be taken once a month, together with a placebo drug for FTC TDF to be taken daily. And then the other arm will take the FTC TDF daily pill, which is an active one, together with the MK-8527, which is a placebo. So when you sit down and ask yourself, these people are at a high risk of getting HIV, and you are giving them placebos. So the question that will arise is that there is a placebo in both arms just to mask and bring the scientific effect. And the evaluation is done on the standard PrEP treatment, which is the Truvada. Sorry, someone is saying I'm moving fast. Can I slow down further?
Yes, maybe
Okay.
just a bit so that some of the audience can catch up, but so far so good.
Yeah, you know, these things are offered, so let us go slowly now. So in this trial, we mean that you are taking the interventional drug, which is the MK, plus the placebo for FTC. We are masking that so that no participant can know in which arm they are, and they cannot know whether they are for the MK, which is taken once a month comfortably, or which is taken or the FTC that is taken on a daily basis. So that crossing now brings in the science, brings in the masking effect, and the fact that you can come to the clinic today and get an FTC active versus a placebo of MK is quite interesting in terms of randomization. Further to this, I can also say we also do randomized controlled trials on standard of care. Like if can go back to TB trial, a TB trial that was done by most of us was on a new drug for TB called rifampicin versus the standard of care, which is the RHZE. I believe you know the RHZE regimen that is provided by GOK for our TB patients. So on one arm, we had the rifampicin guys, and on the other arm, we had the RHZD guys. And further, to the rifampicin arm, we had those who are receiving 10 milligram per kg of the rifampicin, and others were receiving the 15 milligram per kg of the rifampicin, and others were receiving again 20 milligram per kg of the rifampicin. So you could be randomized into any of the 4 regimens within the trial on a 1-to-1, like just flipping a coin, and we get your randomization. So that is how the randomization controlled trials are done. Just flip of a coin, you get an outcome, and that outcome is what the patient is assigned. And you should note further, if you are evaluating a vaccine that is also new in the market, like let's say, for example, the COVID vaccine trial we did just the other year when COVID was around, we gave some participants the actual vaccine, which is the intervention, and others got the normal saline, which was the control group. So in this case, you are able to tell for those who received the normal saline, you could see their sickness level or sickness parameters moving on faster than those who received an intervention. And that was later on migrated to what we call an adaptive design. So we can move next and talk about the adaptive design. So in adaptive design, we are saying, in the adaptive design, we are saying that we have agents that are added and dropped out of the study for efficient testing of the new agents. So in adaptive design, we'll give you a placebo and we'll give you the actual thing. When we see the actual thing is working, we migrate you as a patient who is on the placebo to the active drug. As we move higher the level of active drugs and agents that we have. This is so because adaptive design will allow you to get questions answered very fast. Especially this adaptive design was used during the COVID-19 session. For example, we did a study called the Adaptive Platform Treatment for Trial for outpatient with COVID-19. So we just wanted you to test positive for COVID, that is an inclusion criteria, And as you testing positive for COVID, it means you are an outpatient who is able to move along with your daily chores without the disease putting you down. So in this case, you'd come to the site, you get randomized either to the active drug, which at that time was SAB 185, or the placebo to SAB 185. So as we move along, we would able to tell that as an outpatient of COVID, we would see you having more signs of COVID, and this would be documented, and you would come back again to the site and get randomized now to the actual drug for COVID, and you're given that. While those who were on active drug initially while you are receiving the placebo are migrated into another drug for that purpose. We did that until when we came and discovered that one of the agents that was called casirivimab plus indevimab was active against COVID. So it became our standard treatment. So we could use casirivimab and indevimab on X number of patients while comparing the SAB 185 also on X number of patients. So at that point, you see we have dropped the normal saline. So we are giving you something that is active against the disease. Then if I'm to talk about the pragmatic design, these are normally put under routine care conditions, and in this case, in phase 4 trials and post-marketing research. So, for example, we have a pragmatic design trial for optimized RSV. RSV stands for respiratory syncytial virus vaccine in real world. And this, the mother who is pregnant will come to the ANC clinic, consented, and when they accept, they receive the vaccine. And on the flip side, the other mother who is also pregnant will come to the clinic, consented, and if they accept, they will be randomized to an active placebo drug, to a placebo. So, and that happens at the week 32 of being pregnant, and then such mothers will be followed until their children are around 3 to 4 years. So during this period, we are able to follow these mothers and document the outcome of the pregnancy was safe, and the infant born maybe has suffered respiratory diseases of whatever nature twice or 3 times, and this one who also received no intervention has not suffered or has suffered. This is very important for us as pharmacists because the outcomes of the pragmatic optimized research designs will help us formulate policy. For example, the malaria vaccine that is being used, we can share it on a pragmatic platform and say, those who receive the vaccine were able not to get malaria. Those who received the vaccine were prevented of malaria. Those who did not receive got malaria. So in this way, we are able to inform policy so that with time, the evidence generated can be taken up and we have the malaria vaccine included in routine schedule of KP so that children born within that period and they fall within that age, they are able to get the vaccine routinely just for prevention purposes. So we can move to the next slide. So observational study designs do not really apply much in our clinical trial phases or in our clinical trial setup. So cohort studies, they follow defined groups over time, comparing those who are exposed versus those who are not exposed. Like you can have a study titled Incidence of Cervical Cancer and Associated Risk Factors among women living with HIV compared with HIV negative women in Siaya. So in this, you have a cohort of those who are living with HIV and a cohort of those who are not living with HIV. and you are trying to gauge what is the incidence and what is the other associated factors. So for case control studies, you also start with outcomes. Those are cases versus the controls. And you look for exposure differences. This is efficient for rare outcomes that you expect to conduct. Like you can do a risk factor for TB among people living with HIV in Kisumu, Kenya. So in this case control, you have all people having HIV, but within these HIV guys, you're also targeting who will develop TB within the HIV setup so that you can case control. And then the cross-sectional studies, you just measure exposure and outcome at a single point in time. You like do a snapshot. And an example is you can do a study on antimicrobial prescribing practices around Nairobi community pharmacies. So you can target like 10, 20, 30 pharmacies that do prescribe this, and then you can do a snapshot. Next. So we also come to ethical and regulatory pathway. So as I had initially told you that when you get a protocol from a sponsor, you need to know this protocol is in which design, this protocol, is it a clinical trial? This protocol, does it qualify for evaluation? Does it have human subjects? Does it have an intervention? So gauging on that, the first journey you have to take is to go to the ethics review. So the ethics review, there are some dossiers that you'll have to submit to the ethical bodies for you to get the protocol done. And these dossiers will have same characteristics and composition of documents similar to the ones we submit to Pharmacy and Poisons Board. And basically, ethics review might take 6 to 8 weeks approximately, so that you now move to the next, which is Pharmacy and Poisons Board review. And this one you submit to the clinical trials panel. And these Pharmacy and Poisons Board have a better way of doing things, whereby you log into a portal and submit the dossier. We will talk about what these dossiers are about in the next slide. So while doing ethics review, it has to be approved for it to move to PPB review. So the trick that nowadays we normally try to put in is that when we go for PPB review, we also go for NACOSTI research license, whereby when PPB completes its reviews, they will give you the approval. And that approval, you just plug it in NACOSTI, and NACOSTI will take close to a week and give you a response. Also, they have approved. So for PPB, it might take 8 to 12 weeks or more, depending on the response the reviewers give you and how you respond to those responses. For investigator-initiated studies, to my experience with Pharmacy and Poisons Board, they have gone up to around 6 months. That is because most of our investigator-initiated trials do not have those prerequisite documents required by the Pharmacy and Poisons Board. And to this also, I would urge you to go to the PPB website and also download the guidelines for clinical trials for 2026 as shaped out by PPB. They'll give you more insight to what you're supposed to do. So as I've said, NACOSTI license will have to come out. If you are brave enough to shorten the time, you do all the application together. Then the CHMT authorization, basically we do it within the counties. Like if our trial site is going to be at Kisumu, we'll approach the CHMT for Kisumu for an approval. And this approval will always be granted to you if you also produce the ethic review, the PPB review, and the NACOSTI license, and say now you have cleared with these bodies, and here it is, you're looking for the CHMT authorization. It takes around 1 to 2 weeks, and then you're good to start with your trial. So we can move next and see the kind of documents. Oh, sorry. These are the ethical review bodies that are there in Kenya. This is just a short list whereby, if possible, you can ensure that you go back and like just do a Google search. The one that is known much is the Cambridge Scientific Ethics Review Unit. That's the CERU. We also have the UON, KNH, ERC, the Moi University, the AMREF, the Aga Khan, KU, Ethics Review Committee, Maseno, ERC, Strathmore University, ISREC. And then you come to realize that ISREC is for major referral hospitals, like also Jaramogi Oginga Odinga Kisii Referral Hospital. So these bodies is where you will first submit your ethical dossier for consideration and approval. Then in the next slide, we can talk about the dossiers that you submit to PPB. So as I had said, you have your protocol. So you have to have your protocol synopsis, investigator's brochure, or package inserts. Investigator brochure gives like the pre-analytical phases whereby that drug was developed and given the name it is given or given the code name it is given. Then you have to produce the informed consent for participants, and normally we call them participant-facing materials. You have informed consent, you have brochures and stuff like that. So that can be produced at this point. Then clinical trial application submission form, there's a checklist the PPB has, and that is why I urge you to go take a read on the guidelines. And regulatory authority approval or submission documentation. This is the ethical committee submission, where you get the ethical committee approval. Pharmacy IMP dossier talks about the manufacturing, the characteristics and the summary of medicinal product characteristics about the IP you are producing. Certificate of analysis, the labeling and the packaging information. Also, they will require you to produce pictures of the IP. Then they will require also to know the randomization and blinding procedures. Randomization does just which arm and which arm. Blinding procedures, when we say a study is a double blind, we mean that the treating clinician does not know and the participant who is receiving the intervention do not know which intervention they are getting. So working within the pharmacy in a clinical trial, you are able to tell this one is getting a placebo, this one getting the intervention, or this one is getting the standard of care. So those things come into play. And as a pharmacist, that is very crucial for you. You should not like tell the treating clinicians that so and so is getting a placebo. Now that's when you are opening room for bias. So you also have to produce data management plan, statistical analysis plan, and a monitoring plan. And you have said monitoring plan, these are assisted from the CROs. They'll give you a plan. You also have to provide site staff CV and GCP certificates. I know Dr. Koba will tell you where you are going to train on this GCP certificate. The staff CV are there so that we can see, according to the Pharmacy and Poisons Board guideline, do you qualify to be an investigator or do you qualify to be a sub-investigator? Also, the study budget is and contract agreement between the sponsor and the site. And the budget is also important so that PPB can know whom are we like dealing with and are they able to indemnify participants just in case. So recruitment material and advertisements, laboratory manual, biological specimen collection, storage, transfer plan. These are what we call MTAs. Basically, MTA, material transfer agreements, are done for studies where we collect samples here in Kenya, and these samples are to be shipped, let's say, to Belgium, to a lab in Belgium for some specialized testing that is not able to be conducted in Kenya. Or these samples that are collected here are going to be shipped to that lab in Belgium because the sponsor prefers that to be his referral lab. So that MTA is going to be given to you from the ethical body. And from experience, it takes around 3 months and it expires. So you have to like renew it every 3 months. So data protection confidentiality documentation also should be there. Community engagement plan, where applicable. This is how your guys from the site will go to look for these participants. Are we looking for participants within the community? Are we looking for participants within our hospitals? And how are we going to talk to them? What are the talking points that we are creating for the study? So that when you tell a patient who is suffering from TB that, yes, you're suffering from TB, we acknowledge it has tested positive. We want you to attend this clinical trial. It has benefits A, B, C, D. Those are the things contained in the engagement plan, and how you engage with stakeholders. Stakeholders in this case will be the CHMT that has allowed you to recruit from a given hospital, and then the hospital management committee. And apart from that, there is a CAB, Community Advisory Board members, who come into play, and when they get to know about the study, they can spread the word and tell you that there's a good study here, it can manage this condition of yours. Then for compensation and reimbursement, these are contained in the consent form, whereby we say if you come for procedures to our site, we'll reimburse your transport at 1,000 shillings, and we'll reimburse your time at, let's say, 2,000 Kenya shillings. So when you leave the site and you have done procedures, you have participated in this trial, every visit we compensate you that, because sometimes it's difficult to get a participant just to come to the clinic and you just let them go like that. How best can we say they came, and how best can we support also the participants to come? So study related SOPs, these are basically the standard operating procedures, and majorly if a trial is on site, we would also like to see what we call study specific procedures. These are procedures that specifically talk about that study and how you are going to do your day-to-day execution of work. Trial registration information. So this one, you have to log into something called the South African something Pan-African Trial Registry. You log in there, you give details about the protocol that you're going to do, and then you get that certificate from those guys in South Africa. That is also essential because you are putting that trial in a registry whereby we are able to come back in future and refer. Then any applicable amendments, in case there are amendments coming in form of clarification memos, letters of amendment, you should also be able to provide that for PPB. And this list also is the same list you'll produce for the ethical committee. So this is the dossier in a nutshell. So we can move next. So after you have done your clinical trial, we want to see, does this clinical trial drug qualify to be what it should be in real life? So as I've said before, your site will receive some paperwork, do feasibility, and feasibility is just a list just to give the address of the site, who is the PI, their qualifications, their expertise, their experience in running trials. Then the site will be selected, and this is done by a CRO. Site initiation and startup, also this is done by a CRO. So you obtain approval, you train the staff, you ensure the infrastructure is there. If you have equipment, ensure they are well calibrated and documentation is there. Staff have got CVs, trained on the protocol and stuff like that. Then you hit the ground. And for initiation startup, you should get all approvals. Then you go to recruitment. Recruitment, which cohort are you looking for? Are you looking for TB patient, HIV patient? Are you looking for, let's say, a vaccine trial? Are you looking for who? How do we call them? Healthy volunteers. Then you screen them. Screening is just to assess the inclusion and exclusion criteria according to the protocol. If the protocol exclusion criteria says a lab value of XYZ is met, then the participant is included or excluded. Randomization, as we have seen, is like the flip of a coin, head or tail. Then follow-up. Follow-up will be basically, we are looking at those monthly visits or scheduled visit within the protocol. If the protocol says you'll come every month for this XY safety visit, we want the participant to come. Then exit of the participant will have to take place. That is at the end of the protocol. Then the protocol goes to statistical analysis. So in statistical analysis, data is cleaned, locked, analyzed, and used to answer these objectives. Then there's write-up of manuscripts, whereby you write these manuscripts for publication. And in most cases, wherever I've been, I've seen those who write manuscripts are told there's a conference XYZ, you travel there, you present your manuscripts. And such conferences also give you they give you the transport going and coming back. Like we had the IAS conference happening in Rio. I know of a couple of friends who went there to present their manuscripts. Then we do reports. We comprehensively do the study reports and submit it to stakeholders and regulators. So in number 12, whereby our Ministry of Health, they will review that evidence and they'll engage stakeholders and translate these results into guidelines. And if there are guidelines now, there are strategies and health policies to improve on population health. For example, the RTS MAL 054 was successful and it passed all these steps. And after passing that, it was included as a guide within KP that children born of this age X to Y are able to get the malaria vaccine. Thus, it is being provided as a malaria vaccine routinely within our KP coverage. Next. So in takeaways and discussion, you should think about research is the evidence engine. The drug you prescribe, the drug you dispense, the drug you hold and store under good GMP, whatever practices, that comes from research. Then you should also learn that when you get a protocol, you should be able to match the design to the question. If it is a randomized control trial, if it is adaptive where you add and drop agents, is it a pragmatic one in phase 4? We are looking at RSV to be registered and to be included for maternal vaccination. Is it observational where we are looking for these cohorts? And the most important part now as a pharmacist, in regulatory practice, you should know which ethical committee works for you because there are a number. After that, go to the Pharmacy and Poisons Board. If you are at the Pharmacy and Poisons Board, there is a clinical trial portal. There is a guidance to clinical trial. Get those manuals. Get to know how this is done. NACOSTI, you can also check on their website. It's a government entity. Get to know what are the ethical things you need to follow in dossier preparation to these two. The CHMT, these are your county governments. Wherever you're going to do a trial, will you get access to facility X, Y, and Z? So with that small and long talk, I would like to stop there and return it to Daktari. Thank you.
Thank you very much, Daktari, for your very informative segment. For the audience, we'll be taking questions at the end. For now, allow me to introduce our next speaker, Dr. Kwoba Mangeni. He is a trial pharmacist who also serves as a regulatory officer at the Kenya Medical Research Institute Center for Microbiology Research, popularly referred to as KEMRI CMR, Kargeno Research and Policy Hub in Kisumu. He is a mentee with the African Young Scientists Program and a member of Cohort 9 of the Pharmacist Leadership Stimulant Program out of Nigeria. Driven by a passion for translating formidable research outcomes into policy, he aspires to rise through the ranks to become a clinical trials manager. Daktari, the floor is yours. Karibu.
Thank you very much, Dr. Victor. I will be taking us through the next segment of clinical research. And if I'm fast, please feel free to tell me to slow down. So over the next few minutes, we'll be talking about good clinical practice. We will be talking about the history of good clinical practice, the roles of a pharmacist in a clinical trial setup, as well as how you can plug in to clinical research. So prior to establishment of good clinical practice, there were really ethical concerns regarding how clinical trials were being done. They were being done with no regard to ethical principles, and that is what led to the formation of the good clinical practice. Countries that came up with the formation of the GCP were the founding members were the European Union, Japan, USA, and the big pharma. They sat down and then they decided to come up with the International Council for Harmonization. That is why now in clinical trials it is ICH GCP. So ICH GCP basically operationalizes the ethical basis of research that are contained in the Belmont report. The Belmont report came up with the principles for ethical research, that is respect for persons, beneficence, justice. Respect for persons basically means that you respect the participant, their willingness to participate in the study or not. Beneficence is that the trial that you're doing actually is of benefit to the participant, and justice is actually your fair in terms of how you are tending to this participant. So GCP is these are the standards for the design, conduct, and reporting of clinical studies involving human participants. In a blanket term, it's basically a rule book for the running of clinical trials. And it's majorly focused on two things, that is the rights and well-being of the participants, where they ensure that the right safety and well-being of participants is maintained, as well as data reliability. That means that the data that you are collecting during the course of the clinical trial is reliable and it follows the Al-Kawasi principles. So it, as I've said before, it operationalizes the ethical ideals of the Belmont Report and has been established by the International Council for Harmonization. So it applies to everyone involved in in a clinical trial. So it doesn't necessarily limit itself to only the sponsors. It covers the sponsors, the investigators, ethics committees, as well as the site staff who are engaging in this trial to ensure that the results they get are actually reliable. So it spans every phase of a trial, that is phase 1, phase 2, phase 3, and also phase 4, and from design all the way through final reporting. So in Kenya, as we all know, compliance is enforced by the Pharmacy and Poisons Board. Next slide. So what is the history of ICH GCP? As I have said, many countries were having their own, they had their own different rules of how they were running their trials. So these are 4 countries, they sat down and then they came up with the ICH GCP in 1996. So the first one is the ICH E6R1, which came into effect in 1996. It laid down the 13 principles of ICH GCP, whereby it has 13 core principles, such as ethical principles that have to be followed by the people conducting the trial. It something to do with informed consent, the scientific background of the study is supposed to be sound, amongst other things. I will not go into details on the 13 core principles. I can share a link for you to just go through the document. And then in 2016, they sat down again and then they saw that there were advancements in technology. So from E6R1 to E6R2, they were looking more towards the aspect of risk-based monitoring, where the CRA provides more focus on high-risk sites and also high-quality data. And then they were shifting to computerized systems during this time. You remember before before 2016, they were still using physical case report forms. So the shift to R2 came up with the basis of being able to use computerized systems such as electronic case report forms, electronic trial master file. It allowed also electronic signatures. So it was majorly incorporating the advancement in technology. So for the 2016 ICH E6R2, they were still maintaining the 13 core principles. And then in 2025 now, we shifted to the ICH E6R3. So the ICH E6R3, basically now it was more of a patient-centered, and that means that trials now had to consider the participant burden, and they were now the trial sites are being given the opportunity to consider home visits, to consider remote consents. I know there's a study that we were about to do that now was allowing us to use now electronic consent forms instead of the manual ones where the participants have to come to site. And then it shifted towards decentralizing clinical trials where now, because of advancements in technology, things like telemedicine now can be applied during the course of the trial. And then there was it shifted more on clarification of responsibilities for sponsors as well as the research personnel. And it was also it also emphasizes on data protection and privacy. So for the ICH E6R3, it is now more of a simple, it has a simpler language and it is more understandable compared to the E6R2. So right now it is the ICH E6R3 that is in effect, and that is what now is currently governing the clinical trials. Next slide. So now that we have looked at ICH GCP, what now are the roles of a pharmacist in clinical research? The roles of a pharmacist in clinical research span from IMP management all the way through to quality control. So for the IMP management, this is the investigational medicinal product management. The pharmacist is involved in shipment of the same from the sponsor to site. So you'll have to utilize the Trade portal to do the importation. So they're the ones charged with the generation of the import permit. There's the aspect of receipt of the IMP. So for the IMP, you'll find that majority of them have varying degrees of storage. So there are some which are stored in ultra low freezers. There are some which are stored in fridges. That is a range of 2 to 8 or maybe -40. There are some which are stored at room temperature. So the ranges can range from 15 to 25. So for these IMP, even as they're being transported, they're usually accompanied with something we call a data logger that is able to just track and show how the temperature has been during transit. So in the event that you receive the product and the temperature is out of range, it is usually advisable that you quarantine the product, then you notify the sponsor, and then the sponsor is able to provide more feedback on the same. And then there's the aspect of dispensing. This is done in liaison now with the whole team. The participant will go through the randomization, and then after the randomization, they're able to be assigned to different arms of the study, and then now pharmacy will dispense with regard to what the IRT has dispensed has assigned to the participant. Then there's the aspect of accountability and distraction. So accountability, basically, you're trying to reconcile what you have received vis-a-vis what you currently have in stock and what you have dispensed. So it's usually a very key aspect of IMP management because any discrepancies can really warrant even a stoppage of the trial. So distraction also is also part of the pharmacist's role. Once the sponsor provides guidance and approval for destruction of the IMP, whether used or unused, it is the responsibility of the pharmacist now to take it up and to ensure that it is done. The next role is patient counseling. So even as you are dispensing this IMP to the participant, it is your responsibility as a pharmacist to inform them of any side effects that they may experience, to inform them of how the drug works, so that they are able to, just get hands-on and understand what the trial is all about. Then there's the aspect of pharmacovigilance, monitoring and detecting and reporting adverse events. So these ones are usually done on the clinical trial portals. There's a CTR, a different portal for PPB. So we usually report them both to the ethics committee and also to PPB and also to the sponsor. So they are usually gradable from grade 1 all the way to grade 4. And for the PPB, there is usually a CIOMS form that you fill. So we report them in terms of adverse events or severe adverse events. So they may or may not be related to the study product. And then next, as I have said before, on GCP and regulatory compliance. So it is the responsibility of the pharmacist to ensure that the trial is being conducted in accordance to GCP, as any deviation from GCP will warrant the results unverifiable and they will not be acceptable. And then the next role is data entry. Data entry, I've talked about electronic case report forms. So once you collect this data, we collect it on source docs. Source documentation are basically just like physical, say, paper. And then now you have to translate them now to electronic form. So you enter them to the different databases that the sponsor provides. There are various databases. We have data capture, we have inform, and we have rave ecrf. So there are very many categories of the same. So it also depends with what the sponsor provides. So after you have collected your data on source documents, you transfer it now to the ecrf. Then there's the aspect of quality control. Quality control is basically verifying that processes that have been done throughout the course of dispensation, all through the closure of the study, they meet the study and quality standards. So these are just a bunch. These are just a few examples of the roles of pharmacists in clinical research. They are more, and they also vary from organization to organization. And the golden point is that pharmacy is usually the heart of any clinical trial center. If you go to any clinical trial center, it is the pharmacy which is usually key, because any lapse in monitoring, any lapse in temperature, any lapse in documentation can ultimately result in a pause of a study or closure of a clinical trial site. And additionally, PPB has also strengthened the role of a pharmacist in clinical research. Right now, even as you submit documents to PPB for review and approval of a clinical trials, it is mandatory for the PI to submit documentation of the pharmacist as well. So next slide. So now that we have seen the roles of a clinical research pharmacist, so what are the career pathways in clinical research? So you can venture into clinical operations. For clinical operations, you can become a clinical research coordinator. Then you rise through the ranks to become a senior clinical research coordinator. Clinical research coordinator basically just ensures that they coordinate, even from the term coordinator. They coordinate the study to ensure that the timelines are met, the recruitment is on point, and the necessary documentation is in place, and staff are trained, and also to ensure that the trial is compliant. Then there's the aspect of CTA to CRA to senior or lead CRA. So for the CTA is more of a clinical trial associate. So before it is a stepping stone to becoming a CRA. A CRA is a clinical research associate. Mostly we refer to them as monitors, and they usually monitor the trial on behalf of the sponsor, just to ensure that everything is being conducted in accordance to GCP and that compliance is maintained. So for the CRA bit, I know UN in collaboration with Cambridge do run programs on training of people to become CRAs. It is a program that mostly nurses have taken up, and it is a gap for pharmacists that they can take up. Aga Khan University Hospital, usually their Center of Clinical Research also have programs on training of CRAs. And then there's a CRA school in Montreal, Canada, where they are able to train you on various aspects of becoming a monitor. There's the aspect of becoming a certified clinical research professional, that is the CCRP, the aspect of becoming the CRA, then clinical data manager, and then clinical site manager. So for the CRA school in Montreal, Canada, it's usually online, it's remote. So learning for the program is usually self-paced, and it's usually taken for about 6 months. After that, they're able to give you like a remote internship, so that you can be able to just apply whatever it is that you have learned. And then I've talked about clinical trial manager is basically the one that the sponsor has delegated to oversee a given trial site in a given country. And then clinical operations manager, they're the ones now who are involved in the general operations of the things to do with the shipping of the investigational medicinal product to site, and generally just how things run throughout the clinical research period. And then there's the aspect of clinical pharmacy and supply. So basically, for this, all these roles just lead down to becoming a research pharmacist, where you become the key point person. Jerry has talked about being a pharmacist of record. So you become the pharmacist of record for the site. So anything pertaining pharmacy, it is you become the point person. Then for regulatory and quality, regulatory affairs for a clinical trial site is not like the normal regulatory affairs for that we keep on hearing about. For a clinical research site, it is basically more of ensuring that staff are trained on GCP and they are compliant, ensuring that their the documentation is up to date, ensuring that all the approvals that are necessary for running of the clinical trial are up to date and current, and ensuring that they are renewed on date, etc., etc. And in addition to that, you can also become a GCP or quality officer. I've talked about that being also part of the role of regulatory. Clinical quality manager, ensuring that now documentation that is being collected by the site meets the acceptable quality and is able to be reproducible. You can also become a GCP inspector or an auditor for the site, just to ensure that there are no lapses, that even when you're conducting monitoring visits are being conducted, that the findings are minimal. And you can also join PPB ECCT membership. ECCT is the expert committee on clinical trials. So they're the ones who review the applications that we send to them with regard to approval for a clinical trial. So they sit down, they review, and then they're able to give feedback on the dossier, as well as to give feedback when you are doing renewals. You can also become a drug safety officer with regards now to clinical trials. Now, these are more of a role that is more linked to now the sponsors, that is the pharmaceutical companies. So they are able to just incorporate you as a drug safety officer, and with that you'll be able to work on pharmacovigilance and also safety with regard to the IMP. And then for data and scientific, you can become a clinical data manager. You can be analyzing the data on your side. For clinical research, for a clinical trial, there are some which are blinded, some are unblinded. So for the clinical data manager, there are some also who are blinded, some who are unblinded. So you can be able to switch to either role. You can be on either role, and you can be able to provide data that can inform progress of the study, as well as you can become a biostatistician. So you can be able to collect the information that now the sites are filling on their ECRFs. You are able to tabulate, you are able to come up with decisions that can inform change of the running of the trial. And then for medical and academic research, I believe most of us are familiar with this. This one, you can still upgrade and rise through the ranks and become a research scientist or a principal investigator, like my learned fellow, Dr. Jerry. And you can also become a medical science liaison or a research scientist. So basically, these are the few career pathways in clinical research. These are not all, but these are the major ones that are there, that we feel like a pharmacist can fill the gap, and where more plugin is needed. So it is usually not a case that you just have to believe in yourself. You take up an entry role, and then as time goes by, you're able to progress to higher ranks. Next slide. Next slide. With this, so what's next? So once we have talked about the ICH GCP, we have talked about the roles of a pharmacist in clinical research, we have talked about the career gaps. So what next? You don't really need to have everything put together. You don't need to have like 10 years of experience. You just need to have a starting point. So for instance, you can start by getting your GCP certification. GCP certification, there are a lot of online platforms which offer GCP. There's the CITI program, there's NIDA, there's Clinical Trials Network. So this going through the GCP certification will just help you to understand really how clinical trials are carried out and what is needed of you, and then know the basic principles. That is the 11 core principles of the ICH GCP. So it is a whole guideline. It's about 54 pages. Just go through it to familiarize yourself with what is required, even as you go through clinical research, and what is the expectation. And that will also help you to contribute to ensuring that the data collected follows the Alcoa-C principles. Then build theoretical knowledge, understand research frameworks and expectations of a research pharmacist. Aside from the roles that I have stated, you can just still go deeper and find out what really is needed of you as a pharmacist in clinical research. You cannot only limit yourself now to the Kenyan space. You can also go and just read the WHO recommendations, all these things, so as to just build that theoretical knowledge for yourself. And then learn the PPB guidelines. As Dr. Jerry has stated, PPB came up with new guidelines on the guidance on the conduct of clinical trials. So it is also another booklet around 120 pages or 200 thereabout. I'm not quite sure. So it usually provides the framework of what is needed for a research site. So even as you position yourself to enter into this field, you are able to know what is expected of you. Know what is expected of you with regard to IMP management, with regard to distraction, with regard to shipment, with regards to submission of a dossier for review. So learn the guidelines and then take up internships. Like I have said before, there are some the CRA school gives internships even after you have taken the course, and this will enable you to gain hands-on exposure. I know Camry also sometimes advertises for internships, so it is usually a nice opportunity for you to plug in and just get a head start. They also have this program. They're called Ideal. It is usually for to just inculcate people into research even as they graduate fresh from campus. So that is also a plus. It will also enable you to gain that leverage, and then tweak your CV so that you are able to position yourself for roles in clinical research. You cannot be you cannot have a desire to join clinical research and yet your CV is still reading an industrial pharmacist. You have to so you have to show some initiative that there is something that you have done that leans towards clinical research that it will show even the potential employer that you are willing and you're willing to learn and you are able to take up the role. So I believe that is it from my end. So my key takeaway is that there are many positions, there are many gaps that can be filled by pharmacists. You don't have to know everything. Just start one point at a time and have that desire to know and grow. And with time, you will be able to plug in at any area of clinical research where you feel interested to join. Thank you. Back to you, Dr.
Thank you so much, Daktari, for the wonderful presentation. We've now moved into the Q&A session, and a couple of questions arising from the chats. The first one is it legal to conduct a private research on a registered product, that is, without engaging those regulatory bodies? Maybe we can hear from Dr. Jerry.
Thank you for the question, and my answer will be illegal. You don't just wake up and do research on a product without getting the proper approval. So even if you're talking about private research, of which I consider that to be investigator-initiated research, and the product is also registered, would really like to know this registered product, what other indication can it do? And after we know that, you should be able to place in dossiers for the ethical committee and for the clinical trial process at PPB so that you get it legal. So the answer is it is illegal to do that. And it should not come from us who are pharmacists and know how this pathway should be done.
Thank you very much for that response, Daktari. If you have any other question from the audience, kindly post it in the webinar chat, or you can raise a hand. I see some people are asking about the title of the presentation on the PPB portal. As Dr. Manyala had stated, it has not yet been uploaded, but we believe it will be uploaded soon. As soon as it's up on the website, you will be informed. So look out for that. I see our presenters did such an excellent job that we don't have a lot of questions today. Okay, awesome. If there are no further questions, I believe I'm not locking anyone out. So we can have the closing remarks.
I think we have 3 questions in the Q&A.
Okay.
I think the first one is whether it is legal to conduct a private research on a registered product without engaging the regulatory bodies. That's from Isaac Chege.
Yes, that's the question that Dr. Jerry has just answered.
Okay. And then there's how far are we with bioequivalent trials, especially bioanalytical testing?
Yes. So Dr. Koba, Dr. Jerry, if you can kindly inform us how far we are with regard to bioequivalence testing.
From my end, I haven't really quite encountered bioequivalent testing yet, but perhaps Dr. Jerry has encountered some. He can maybe shed light on it.
Hi everyone, I can be for this bioanalytical testing. I haven't had any much stories. The last time I heard is that we were looking for how to come up with the bioanalytical laboratory to handle these specimens. Thank you.
Okay, thank you for that response. Maybe I see our treasurer, Dr. Sami Masibo, is in the house. Maybe he can shed some more light on that.
Not really. I'm also not very familiar with that question. So yes, I can't add anything. Perhaps my question would be to either of them. Much of our evidence in terms of our clinical guidelines usually comes from studies that have been conducted externally, most of the time conducted within high income countries. So I wanted just to ask either of you, because even when there's evidence generated within Kenya or internally within Africa even, it often requires external validation before being accepted by even our own countries. What contributes to this, such that we do not really believe our evidence, and how can we close the evidence to policy gap, especially when it comes to local evidence?
Yes, that's a good question. Over to you, our speakers.
Basically, I think from my end, we just have to be more rigorous, and that will enable us to close that gap. For instance, I know there is a study that we are in the process of closing out on HPV, cervical cancer. In as much as the study has been conducted here in Kenya, it has informed global policies, and we are currently even in the process of adapting one single dose of HPV to prevent cervical cancer, as compared to the standard guideline for the MOH, which is the 2 doses. And then we have also been able to, just extend this vaccine, not necessarily to the limit of 9 to 12 years. It can now be injected to people as old as 60 years. So I believe we are currently on the right path in bridging that gap. We just have to be more rigorous and also position ourselves to as people who are able to generate credible results from our research. I believe also there's also a grading with regard to clinical research. Ghana, I think, received a higher rating of late. So I think Kenya, we still have some way to go so that we can also be able to, to reach that level.
Thank you very much, Daktari. And maybe just to add on that, I believe in summary, it boils down to two things. The first is capacity building. So we need to ask ourselves, do we have the capacity to go that particular direction? And if yes, where are we falling short? If no, how can we bridge that particular gap to ensure that as a country, we are also privy to such privileges? Which reminds me of the second point. It's also a policy issue. For instance, I believe it's Japan, whereby every product registered in that particular market needs to undergo clinical trials, whether it's an innovator brand or a generic brand. But now the problem with such an approach is that it inadvertently raises the cost of medicines, because the reasons why generic products are affordable is they don't have to conduct clinical trials. So it's a conversation that we can continue having, but I agree with both of you. I see the question on bioequivalence has also come up a bit. I'm also not.
I can also add that question.
Okay.
Why those happenings happen? Yes, we say Kenya clinical trial data is considered invalid. This is because of one thing, and Koba can tell you. We are provided systems by the sponsors, and these systems will log in patient information, which is confidential. This system provided by sponsors are not anchored in Kenya. So basically, the problem is that Kenya, for us, we don't have that right to intellectual property, trial ownership of the data that we generate, and also translation of the research into locally developed products. Like let's say today we are seeing some light, some change, Kiasi, whereby we are doing once a month prep bill, and basically I've seen there is also a document out saying that it has worked. And also here in Kenya, we have already gone ahead and got a manufacturer anaitwa nani. Huyu ana manufacturer hapa Limuru anaitwa nani. There is a name here, is forgetting. They are going to produce that alimentavir. But the problem we have is that we don't have control over financing of such clinical trials. Product development does not take place in Kenya. Intellectual property issues, trial ownership, data analysis. And this is why when we do these trials, most of the data is not in Kenya, but in the sponsor's domain. And this way, we are more deficient like to accept what is ours, and we go ahead and accept what the sponsors are providing. Thank you.
Okay, thank you for that. And also to add, in as much as we say that as a country, we really don't have ownership of our own data, we've also taken significant strides to ensure that we have some control over it. For instance, in the Data Protection Act of 2019, there are certain stipulations regarding cross-border data transfer, particularly for sensitive data, which healthcare information is classified as sensitive data. And one of those stipulations is the country that is receiving or handling that particular information needs to have equivalent or adequate regulations to ensure that privacy is upheld. So I think it's a matter of baby steps. Regarding the issue of bioequivalence, I'm also not really an expert on that, but I believe the situation in the country is that at the moment, it's one of the requirements for us to attain WHO maturity level 3. And PPB has been working closely with WHO to ensure that we get there. As regarding the status update on that, I really can't speak to it because I'm not privy to that particular information, but maybe it's something that we can look into as a committee and handle it better or have a session around it as well. And then there is a question on an impact research or any other site enter into agreements with the lab to offer bioequivalence locally.
Yes. For that question, I think impact research is an open site as long as we have those contractual agreements with the BE, and that will be a welcome point for us. We also welcome industries to have impact research as their site to do those BEs, because BEs are considered phase 1 trials and we have the capacity. Thank you.
Okay, thank you very much, Daktari. Any other question from the audience? You can just type it out in the webinar chat or post it in the Q&A session. In the meantime, I'd like to invite Dr. Sami Masibo to say one or two things.
Thank you, Doc. I don't really have much to say, but just sharing the excitement when we have such webinars, because I think PSK is trying to open up new. They're not really new, but previously not really exposed career options for pharmacists, and clinical research is one of those things I think a pharmacist will really thrive in. We have had examples of the two presenters who are really thriving in that space. So I'm hoping also we can get many of us in them. Dr. Mangeni, Dr. Jerry, please reach out to those people who might be interested in joining what you're doing, just to guide them, hold their hands, and show them the way around clinical research. It's one of those very rewarding spaces, I think. So thank you so much for offering to take us through what you've taken us through, and even beyond the webinar, just holding hands for especially our younger colleagues who might want to try out some of these things before they decide where they actually belong. I think thank you for the audience as well for coming in and listening to this. Back to you, Victor and Manyala.
Thank you, Bwana Treasurer. So I don't think there are any announcements today. But in the meantime, you can connect with us on our social media pages. I believe they are on your screen. And you can also look out for the next webinar in this particular series. You will be informed about it in due time. Manyala, your hand is up.
Yeah, thank you. Thank you so much, Doc, and everybody else, the panelists, the speakers. And thank you so much, Dr. Masibo, for also just coming in. I wanted to just make a small announcement. I've just been reminded we have an ongoing stakeholder survey, which is to support PSK in coming up with our strategic plan and, of course, refining our constitution. So the survey is ongoing. We have a deadline of Friday, 21st, which is next week. So that's ideally 7 days from now. And we would like you guys to share your inputs and contribute. Your opinions really matter. We've sent reminders, and we will be doing so progressively between now and Friday. So something to look out for. And yeah, thank you. Thank you so much for making time to join us tonight.
Okay. Thank you, Dr. Manyala, and to the panelists and audience as well. Thank you for making time to just come sit in and have this conversation with us. At this juncture, if there is no any other business, consider the meeting adjourned.